Good morning. How is everybody doing? I just want to thank Jan Hodzinski for that nice introduction. Thank Don Ingram and Ann for helping organize much of this meeting. And thank you all very much for coming. So I am from Gainesville, Florida. I'm sure most of you have been to Florida at least once in your life. Gainesville is effectively in the north part of Florida, north central part of Florida, a couple of hours north of Orlando. It's home to the University of Florida, which last time I checked was about 14th as far as academic ranking as far as academics go. The football program, however, is not nearly as good as the academics right now. It's very famous for one thing, and that's Gators or Gatorade. A guy named Robert Cade, who was a chemist, developed Gatorade in the 60s, and it's sort of taken off, and it's a major feather in the cap at the University of Florida. There's a very entrepreneurial spirit also at the University of Florida. Many startup companies and patents are developed out of there. So there's a legacy that Robert Cade developed. The other thing that it's known for, which you might not know, that I think is important, particularly in the context of what I'm going to talk to you about today, is it's home to the McKnight Brain Institute, and also home to what is called the National High Field Magnet Laboratory, which is funded by the National Science Foundation and your taxpayer dollars. It basically looks at brain function, it looks at tissue, and it looks at animal models of various diseases, and it looks at humans and uses high field magnetic resonance imaging to do that. And the other thing that Florida has, which you all know, and you clearly have here in Baton Rouge, which I've seen already, and that is sunshine. In Florida, it attracted our family there a little over 4 years ago. And these are my three daughters, which you see here, looking out over to the ocean. And as I look at my kids and I see them grow, and they are growing as we speak, it reminds me of connecting both how my kids will perceive diseases in the future and how your kids or grandkids now might also perceive diseases in the future. And so what I want to try to do today is tell you a little bit about research in two different ways. Okay? The first way is I want to talk about the future, the way that I think that drugs will be evaluated and developed and tested in the future in the context of Parkinson's disease. Okay? There's, I think, a model that is shifting away from looking at a patient and seeing the response acutely to the medication to looking at the way that a marker may change in response to the medication. I think that is a shift that is gradually occurring, and you will see it gradually occur over time. The second thing I want to talk to you about is the here and now. So the first part may not relate to you now, but it may relate to your kids or grandkids. And the second part can relate to you now, and I want to talk to you a little bit about a specific exercise program which we did in context with a group in Chicago, which showed high efficacy for people with Parkinson's disease. So why study the progression of Parkinson's disease? Well, there's several reasons to do this. One reason is because it's just fundamentally you need to know how the brain is evolving over time, which parts of the brain change, where the pathology is changing over time becomes very important, because if you can learn where it goes in the brain over time, you might have a better ability to stop its progression over time if you know where it's going and it's moving. The second reason is because if you can develop markers or outcome measures of the brain over time, they may be very useful as outcome measures in clinical drug trials in the future. And instead of, say, evaluating the effects of a drug, say many of you are probably familiar with Sinemat or levodopa, you see the effects very acutely. You see the effects within, say, 30 minutes to an hour. The medication kicks in and you move a little bit better. Well, there may be medications that don't work like that. They may modify biological function, but not symptomatic function acutely. And so we need outcome measures that can evaluate that going forward. So this was echoed in a quote by a guy named Warren Olenow. This is in New England Journal of Medicine in 2009. He says, a limiting factor of current clinical trials, is what he's talking about, is the requirement for a clinical endpoint that reliably measures disease progression and is not confounded by the study intervention's effects on symptoms. And so this is a theme that I'm going to sort of keep echoing for the next few minutes. So if you were to look at MRIs today, and many of you probably have had an MRI at some point in your lifetime, you go get an MRI, and if you're a patient with Parkinson's disease, for the most part it's going to tell you, or the MRI is going to come back normal. It's going to say there's nothing there. There's nothing that's obvious. And that's the way the state of the art is currently. Okay? So the clinical MRI today for Parkinson's disease is normal. But what I want to show to you today is a little bit of evidence that suggests that we can actually use current technology and slightly more advanced ways to see things that are not currently seen in clinics. And those particular outcomes may be very useful going forward and into the future. So I don't know if anybody saw this episode. It was recently aired on 60 Minutes. It's an awesome episode. So I would Google it afterwards and look it up on your computer if you get a chance. It was on glioblastoma, and it has no real relation to Parkinson's disease, but I'm going to try to make the link for you because I think it's a fundamental link. And they basically are doing a study at Duke University where they're injecting a polio vaccine in the brain trying to take away a glioblastoma. People with glioblastomas can pass away very quickly after the cancer takes effect. And in this study, the polio vaccine is doing a very good job of sort of eating away at the cancer. And what struck me in watching this episode was not necessarily the drug or the glioblastoma per se, but was the fact that they were using magnetic resonance imaging to see the cancer go away. The patient to them clinically would look the same before and after the polio vaccine. So it wasn't the patient's symptoms that were driving the way that the physician saw that the particular intervention was working. It was the outcome on magnetic resonance imaging where they saw the cancer going away. And by seeing the cancer go away, that told them that that polio vaccine may actually be having a very dramatic effect on this glioblastoma. Another good example, which many of you are probably aware of, are cholesterol-lowering drugs. So statins are typically utilized to lower cholesterol. And basically, you know, if you go get a blood test, your cholesterol is a little bit high, a physician might put you on a statin or a cholesterol-lowering drug, and you go back and see that physician 3 to 6 months later, and you look the same. You know, you're just as strong, you're just as fast, you don't really look that different to the physician. But in this case, though, your cholesterol is lower. And what that tells the physician is that that's going to lower your risk profile for developing heart disease in the future. And so that's really the model that Parkinson's disease is I think, moving towards. It's going to be a slow slog getting there. But I think eventually that this particular disease, if therapies are there and outcome measures are there, will move to that particular model. So I'm going to get deep into science for just a moment. There's a part of the brain called the substantia nigra. It sits, like if this is your brain, and if you come down about like this, it's right in the middle of the brain. And it's an area of the brain that produces dopamine, which is very important for thinking, it's very important for moving, and it's very important for the way you process your emotions. And in this particular part of the brain, the substantia nigra, cells essentially are degenerating over time. And so wouldn't it be nice if we had some sort of measure that might mark or track the progression of the substantia nigra over time? Because most of the medications that are out there right now or are likely going to be developed in the future, many of them are trying to alter the way that the substantia nigra is functioning, or the way that dopamine is produced within the substantia nigra, or the effects that dopamine has outside of the substantia nigra. And so wouldn't it be nice to have a particular marker that might sort of measure this cyclical process that is going on as these dopaminergic cells are changing in people with Parkinson's disease, or potentially even before people get Parkinson's disease? So magnetic resonance imaging was developed in really the 70s, and vendors started using it in making scanners in the 80s. And this is a typical type of scan outcome that you would get, which is what is called a T1 weighted sequence. And as you can see, you can't see everything on this particular image very well, but you can see things on what is called a diffusion MRI sequence, which you can see here. You can start seeing these colors, and these colors relate to the anatomy very well. And diffusion MRI is effectively a technique which is looking at the way in which diffusion molecules occur within tissue and the constraints on that diffusion process. So the best example I can think of is if you take a bowl of water and you put purple dye in that bowl of water. As many of you know, the purple dye is just going to diffuse throughout the water, and the bowl will be purple. Okay? And then basically if you were to put constraints in that bowl of water, like 2 steel beams, and you drop the purple dye between the 2 steel beams, the purple dye would sort of diffuse along the lines of the 2 steel beams. So the constraints in the diffusion process could be measured, and that's basically what diffusion MRI is doing. It's measuring the constraints in the diffusion process. So this is another nice example. If you think of dopaminergic cells in the substantia nigra changing, this is basically a voxel in the brain from diffusion MRI, and each of these little circles is a cell, okay? And inside the cell you have diffusion going on, which is the red, and outside of the cells is the blue. You also have diffusion going on. And this diffusion magnetic resonance imaging is effectively picking up this diffusion process. We think it's more likely picking up the diffusion process outside the cells. So we think it's more likely picking up these blue lines outside of the cells. And another good way to think of this is balloons, okay? So recently my wife took our kids, for some reason, I still haven't figured out why she did this, but she, on April Fool's, filled up a lot of balloons, about 1,000 balloons, and put them in my office. These balloons effectively could be thought of as like cells, okay? And so if you were to pack this room, that room right there, all full of balloons, the balloons wouldn't really move around that much, okay? But if you then popped, say, 50% of those balloons, they might move around a lot more, right? And so the environment outside of those balloons would grow. The volume of that environment outside of those balloons would be growing. And what we think is that this diffusion MRI technique is picking up on the sort of fractional volume outside of these cells in deep areas like the substantia nigra, and what we're trying to do is sort of track them over time. So we essentially use magnetic resonance imaging. This is a typical MRI scanner. This is a Philips scanner, but we've done our technique on GE, and we've also done it on Siemens, and we're getting similar effects on each of these particular systems. And this is the processing pipeline that we use, which is basically how we process the pictures. So taking the pictures is pretty standard, but the way we process the pictures is really the unique part. And what you see here on the far right is the midbrain area, which has the substantia nigra in it. And this is basically what is called a free water map, which is basically measuring the amount of free water content within the midbrain area. And these this is a healthy individual here, and these bright areas in here indicate a slightly elevated levels of free water content within the substantia nigra. And as you see, it's that posterior part, this P area, which has the brightest signal, okay? And we think that's very indicative of the degenerative process going on, or potentially even the inflammatory process going on in Parkinson's disease. So we did a study. Everybody does a study, but we did a study at the University of Florida in a cohort there. And we looked to see how does the free water content in the substantia nigra change in people with Parkinson's disease compared to healthy individuals. And I don't want you to look at every part of this slide. I just want you to focus on the orange here. The orange basically shows you that on the y-axis is the free water, and people with Parkinson's disease have a higher level of free water in the substantia nigra, particularly in the posterior part of the substantia nigra. And that's the part of the substantia nigra that has the most consistent and most abundant loss of dopaminergic cells in people with Parkinson's disease at pathology. And so what that suggests is that we're actually picking up on a part of the brain which is highly representative of a degenerative process going on. And so this is a study of a cohort. It's 28 people and 20 control individuals. And so what we wanted to do was ask the question, can we sort of see anything related to cognitive measures or motor measures, okay? And so this is cross-sectional, so this is not tracking progression, but this is across people. And it gives us a clue that measures of cognition are related to this free water measure in the substantia nigra, so that higher free water means worse cognition. And then this is the Unified Parkinson's Disease Rating Scale, which is a motor test, which many of you, if you've had motor evaluations, they have you tap your fingers or rotate your hands. That's basically the motor evaluation. So the higher the free water in the substantia nigra, the worse the motor symptoms were. So it seems like cross-sectionally, the amount of free water content relates to cognitive, or how you think, and also relates to motor symptoms, or how you move. So it's all well and good to show things at one center or one site, but the next thing we wanted to do was ask the question, can we replicate this finding in a completely independent cohort? And what we decided to do was grab the data from the Michael J. Fox Foundation. They have a very ambitious and I think worthwhile program, which is called the PPMI, the Parkinson's Progressive Marker Initiative. It's following worldwide hundreds and hundreds of PD patients and healthy individuals over time. And one of the things they're acquiring is diffusion MRI. So we acquired that data from them. free of charge, thank you. And then basically ran the analysis on their baseline data, the exact same analysis that I've shown you already. And what we found was, we found the exact same finding, okay, which is the good part. Basically, we found an elevation of free water in the posterior part of the substantia nigra. We found no differences in the other parts of the substantia nigra. So this is a very good finding because this was across 8 different scanners, all Siemens scanners. Ours was a Philips scanner. So it suggests we're getting a similar finding across different vendor platforms. And it suggests that we're getting it in the same part of the substantia nigra, which is, I think, also a very important finding. Okay, so now I'm going to transition to talking a little bit about progression because that's really what I want to tell you a little bit about. And so what we're doing also is following patients, baseline and 1-year imaging. All of this is done off medication. And we're essentially asking the question, can we track changes in the substantia nigra over time? Okay, and if we could do this, then we might have an outcome measure that people could use to test medications on in the future. So basically, this is a caricature of a patient with Parkinson's disease who might progress over time, and it's really just a drawing. You know, it's a cute drawing, and it's really meaningless. The data here you see, this is actually real data, though. So this is the blue area is free water content in the substantia nigra, which is higher. So blue means high free water. And this is a patient after a year later, and you can see the free water content in the substantia nigra is growing. And what that suggests is that, 1, we can visualize it in an individual patient, and 2, if we can visualize it, that means we can also quantify it. And so when we quantify it, in healthy individuals, you can see the data here. So basically, this is the free water content in the substantia nigra. This is baseline and 1 year later. So the first thing we wanted to do was ask the question, do we get the same measurement in a group of healthy people at baseline in 1 year? Because if we don't, then that's a problem. Okay? We need stable measurements over time that are replicated. And you can see no difference in the control group over time in these particular data, which suggests that we have a very stable measure. In contrast, people with Parkinson's disease increased in the substantia nigra, the posterior part of the substantia nigra, after 1 year. And so basically, in science, this is called an interaction. You get an increase here and no change there. And that's very important because it suggests that we're able to track degeneration going on the substantia nigra over time. Now, even more importantly, was that we're able to predict clinical change, both motorically, movements, and also cognition, thinking, over the next year. So the baseline imaging predicted how well patients did the subsequent year. Okay, I'll say that again. The baseline imaging predicted how well patients think the next year and move the next year. And so this is data for baseline measures of free water. It's separated as from sex, males and females. Females are in the open circles. The dark circles are the males. This is baseline free water in the substantia nigra. This is the actual change in bradykinesia, which is basically those movements that people perform. So basically what this says is that the higher the free water at baseline, the faster the decline of the movements that they would have the next year. So the worse off they're going to get the next year, the higher the free water levels. What this suggests is that one might want to have a medication that holds down free water levels in the substantia nigra so that you can slow the rate of subsequent progression over subsequent years. That's the sort of take-home message of that slide. In addition, a slightly weaker finding, but still it was significant, the baseline measures of the substantia nigra here again predicted the actual change in what is called the MoCA, the Montreal Cognitive Assessment. It's basically a general cognitive assessment scale. And those people with higher free water in the substantia nigra had a faster decline in the MoCA the subsequent year. So basically baseline imaging predicts motor and also cognitive changes the subsequent year. So just to summarize where we're at on the sort of future parts of this talk, one is I've shown you evidence that free water is increased in Parkinson's disease at a single site study, and it's replicated across a study in the Michael J. Fox Foundation. I've shown you evidence that free water is increasing over 1 year in people with Parkinson's disease, but not in a healthy control group. And I've shown you evidence that we can use baseline imaging to predict subsequent changes the next year. And what we're doing now, and I'm hoping other people will take advantage of this as well, is planning to evaluate future therapies to slow free water progression in people with Parkinson's disease going forward. So I've talked to you a little bit about the future and how it may affect our kids and grandkids and the way drugs will be developed in the future. But what I want to do now, as I close, is talk to you a little bit about the here and now, what you can do. Okay, and I want to tell you about one study in particular that we ran that was very efficacious as far as some of the motor and cognitive symptoms for people with Parkinson's disease. So I want to talk to you about resistance training. So basically, if you're lifting weights, you might not think that lifting weights, which is good for your muscles, is actually good for your brain. And I think there's a very good argument to make that lifting weights and creating muscle forces with your body is actually very good for your brain. So we've done a lot of studies in healthy people where we do functional magnetic resonance imaging, and what we found is that as people produce forces, higher and higher and higher forces, similar to what you're seeing in this video here, we see activity across a big network of areas of the brain, and we see increases in blood oxygenation in areas of the basal ganglia and areas of the substantia nigra, which are very important for degeneration in Parkinson's disease. So if basically contracting muscles of your hand increases blood flow to those areas, then imagine what it does when you do a whole body workout. It must be doing much more for those parts of the brain and much more benefit to those parts of the brain. So what we decided to do was actually assess this in a very large-scale clinical trial. So we had individuals exercise for 2 years, 24 months, 2 times per week, 90 minutes per session. There was no other exercise going on except resistance training. For 6 months, they had a personal trainer twice a week, and then from 7 months to 24 months, they had a personal trainer once a week. But they still worked out twice a week. Okay, so you're still working out twice a week, 90 minutes each. There were 2 groups. One is doing lifting weights, resistance training, and the other one is doing what is called Fitness Counts, which is through the National Parkinson's Foundation. It is an exercise program, but it's not very intense. The resistance training, I would say, is a slightly more intense program. And so what it involved was the resistance training involved a lot of different upper limb and lower limb exercises. So we wanted to strengthen the upper limb, but also your core muscles and also your lower limb muscles. And so it was really a whole body workout. And all major muscle groups were involved. There were 11 strengthening exercises. And what we tried to do was basically lift the amount of weights that they lifted over time. So if they got stronger, we increased the weights. We pushed them, basically. So this is, I'm going to show you the outcome on what is called the Unified Parkinson's Disease Rating Scale, which is right now the main outcome measure used to test drugs, usually, for symptomatic therapy for Parkinson's disease. And so on the y-axis here is the Unified PD Rating Scale. Up here it means bad. So this means that the symptoms are getting worse. And so this green dot over here is what the typical change would be in people who are not necessarily in an exercise program. They would generally get worse about 2 to 3 points per year. This is the estimates from large-scale studies through the Parkinson's Study Group. If you look at the Fitness Counts program, however, this is what the data look like. They basically get better at 6 months, and then they sort of progress back towards their baseline at 24 months. But what you can see here is that they're still doing better than what the typical progression would be. So they were effectively able to hold their symptoms constant for 2 years. Okay? All these evaluations are done off medication, so this is not really due to the changing medications that could be occurring. This is all evaluated off medication. Now, in contrast, though, the resistance training group did better. Okay, they got better just like the Fitness Counts group did initially at 6 months, and then they were able to maintain that benefit over a 24-month or a 2-year period. So you can see this is potentially a very beneficial program, and whenever you get over about a 5-point change in the UPDRS, it becomes a clinically meaningful change as well for most people's estimates. So we also assessed cognition in that study. We had several different cognitive tests, which this data was just recently published. So this is basically tests of memory. It's tests of inhibiting one's thoughts, trying to do something regular and then stopping yourself from doing it, and then basically also an attention type task. And in each of those tests, both at 12 months and at 24 months, each of these exercise programs did better. So you can see the blue means better. So all these data points are above the baseline. In each of these tests, they're all significantly higher, suggesting that any exercise program is helping cognition in this particular study. So anything you do in this study was helping the way you think or you memorize or you attend to stimuli. So just to summarize the second part, and as I close, I've shown you evidence that resistance training, which is basically lifting weights, has shown the longest and it's the most efficacious clinical efficacy out there over the longest period of time in a 2 randomized clinical trial. I've also shown you evidence that both resistance training and cognition and fitness counts also increase cognition over a 2-year period, and they were sort of equally effective in that particular study. I just want to thank a number of people in our laboratory. I have a lot of great colleagues that I'm fortunate to work with. I want to thank funding sources through the National Institutes of Health. We also are funded by a couple of foundations, the Tyler's Hope Foundation and the Bachman Strauss Foundation. I also want to thank you all very much for your attention.